So here we will discuss about the pyrimidine metabolism, the biosynthesis of pyrimidine and its degradation. So pyrimidine synthesis via the de novo biosynthesis. Here there is a difference with that of purine biosynthesis, because unlike the synthesis of purine nucleotides, the six-membered pyrimidine ring is first formed and then it is attached to the ribose 5-phosphate. So this makes the basic difference with that of purine nucleotide, where the purine ring is being formed over the PRPP platform. So the pyrimidine nucleotides are as follows, like CMP, UMP, cytidine monophosphate, uridine monophosphate, and thymidine monophosphate. So this is the structure of pyrimidine ring. How it is being numbered: 1, 2, 3, 4, 5, 6. Various elemental constituents are being contributed by various chemicals, like aspartic acid is contributing or furnishing the C4, C5, C6, and N1. So these all four constituent of the ring are being provided by aspartic acid. Then glutamine is contributing to nitrogen 3 and carbon dioxide is contributing to carbon number 2. So this is how, by contribution of these three chemicals, there is a synthesis of de novo synthesis of pyrimidine. So we will see it here one by one. There are overall 6 steps. So in the first step, the carbon dioxide and glutamine, they combine with each other in the presence of enzyme carbamoyl phosphate synthetase type 2. In the presence of ATP, 2 ATPs are utilized and the formation of carbamoyl phosphate. Then aspartate is in the second step. The aspartate transcarbamylase enzyme is there. Aspartate is now contributing to the formation of carbamoyl aspartate. Then in the next step, the dihydroorotase enzyme is there, and this carbamoyl aspartate is now cyclized to dihydroorotate. Further, this dihydroorotate, in the presence of enzyme dihydroorotate dehydrogenase, is converted up into the orotate. Here, one NAD is utilized and converted up into the NADH. Then orotate further converts up into the orotidine monophosphate, the first nucleotide, and PRPP is donating the ribose 5-phosphate and pyrophosphate is released. So this is how OMP is formed, and from OMP there is a formation of uridine monophosphate with the loss of carbon dioxide. So the uridine monophosphate, the nucleotide, the first nucleotide of the RNA is being formed. Then uridine mononucleotide further can be converted up into the UDP in the presence of kinases. Then UDP can be converted up into the UTP. Further, UTP will be able to convert it to CTP, cytosine monophosphate, with the help of glutamine, and the enzyme is CTP synthetase. So this is how the uridine triphosphate can be converted up into the cytosine triphosphate. And further, this UDP, because here the sugar is ribose sugar, so with the simple ribonucleotide reductase enzyme, and reductase require the NADPH. NADPH is converted up into the NADP, and this leads to the reduction of ribose ring, which converts up into the deoxyuridine diphosphate. Then DUDP or the deoxyuridine diphosphate further converted up into the DUMP with the loss of phosphate. Then DUMP can be converted up into the thymidine monophosphate. Very, very important reaction, because here there is an important role of tetrahydrofolate, which is acting as a donor of methynyl group. So one carbon species is being donated by this THF. That's how the UMP is able to be converted into the TMP. Once there is a folate trap due to the deficiency of vitamin B12 or other causes, so that will lock this reaction and there won't be conversion of UMP to TMP. So it has been seen in many types of anemias like megaloblastic anemia. So due to the deficiency of this DTMP, there are immature erythrocytes are there. So this is about how the formation of various these nucleotide, mononucleotides, pyrimidine nucleotides is there. Then about the regulation. So regulation, the first two enzyme, the first enzyme was the carbamoyl phosphate synthetase type 2, and second one was the aspartate transcarbamylase. So both these are allosterically no inhibited by UTP and CTP respectively. And they can be induced by PRP. Excess of PRPP always increase the or facilitate the formation of this pyrimidine nucleotide, and excess ATP also induces the synthesis of purine nucleotide. So these two enzymes are allosterically induced and inhibited by these substituents. So carbamoyl phosphate synthetase 2 is feedback inhibited by UTP and activated by PRPP. Similarly, the aspartate transcarbamylase is feedback through feedback inhibition by CTP and activated by ATP. So this is the trend of regulation. Then catabolism of pyrimidine nucleotide. So end product of pyrimidine nucleotide or catabolism is uh, they are all highly water soluble. They are carbon dioxide, ammonia, beta-alanine, and beta-amino isobutyrate. So humans probably the transaminate the beta-amino isobutyrate to methylmalonic semialdehyde, which is then converted to succinyl coenzyme A via the methylmalonyl coenzyme A, and in the presence of vitamin B12. So this is important intermediate of Krebs cycle. Then beta-alanine can serve as precursor of acetyl coenzyme. So both these products are highly useful in the body. Then disorders, what are the, we can see here the pathway, how there is a degradation, cytosine to uracil with the loss of ammonia, then uracil converts to dihydrouracil. NADPH is utilized here. Then dihydrouracil further converted up into the beta-alanine and with the loss of carbon dioxide. Then similarly, thymine is converted to dihydrothymine. And here there is a loss of beta-amino isobutyrate and there is a formation of ammonia. So we can see here there is a degradation. Then disorders of pyrimidine catabolism. Since the end product of pyrimidine catabolism are highly water soluble, so overproduction of pyrimidine catabolism is very, very rare, and with clinically significant abnormalities. The disorders associated with pyrimidine synthesis are uric aciduria and Reyes syndrome. Thank you. This slide shows the anti-cancer drugs and their target enzymes. So in the pyrimidine nucleotide synthesis pathway, so we can see it here, the fluorodeoxyuridylate. It's inhibiting the thymidylate synthase enzyme. So there is no conversion of UMP to DTMP. So the pyrimidine synthesis is hampered. And here the dihydrofolate reductase is also being competitively inhibited by aminopterin and methotrexate. So both are anti-cancer drug. So this is how the cancerous growth is inhibited by arrestment of these enzyme activity. Thank you.